Can 4-PBA be used as a chaperone to help fold proteins? I believe it can, and I can believe that it can be done safely and effectively at very low doses in tissue culture, in mice, and in humans. In this video, we're specifically going to look at mutated collagen 4A1 and 4A2 proteins and how 4-PBA interacts with them and helps them to fold. But I know what you all are wondering. Can it fold my laundry? You ought to be ashamed of yourself. You could be asking, could it help with Parkinson's disease or Alzheimer's, Huntington's, or Creutzfeldt-Jacobs disease? There's all kinds of unfolded protein diseases out there that it may help with. And I believe that we can help adjust that dose to the perfect dose. This video is a basic video to help prepare you for the next video, which is the unfolded protein response video. In it, I talk about how to adjust a dose based on those markers. Okay, my name is Michael Singleton. I have a master's degree in molecular biology. I'm not a doctor. If you are considering chaperones, please discuss that with your healthcare professional. So in this video, we're going to look at the structure of 4-PBA, how it's polar and nonpolar, and how these areas make hydrophilic and hydrophobic areas of that molecule. Then we'll look at collagen, its structure, the glycine XY repeats, and how this sets up a trimer in the center of that molecule. We'll also look at how 4-PBA could possibly be interacting with the molecule to help it fold. And if it does not fold, we will look at how aggregation happens where they all clump together and gum up the endoplasmic reticulum, making the endoplasmic reticulum give an unfolded protein response. And we can measure that with markers. And when we do, sometimes we can find them in the serum and in the cells. So they can be used in cell culture, in mice, and in humans to find a therapeutic dose. So 4-phenylbutyric acid has a phenyl group on the end, and it has 6 carbons at each angle there. These are double bonds in between. And off of it comes a butane, which is, but means 4, 1, 2, 3, 4 carbons, and has attached to it a carboxylic acid. 4-PBA is also known as sodium 4-phenylbutyrate. That's when the sodium replaces the hydrogen. It comes like that in a powder. When it's dissolved, it separates and makes a solution. In order for you to fully understand how 4-PBA interacts with collagen, I need you to understand how it is polar, what makes it polar, what makes it hydrophilic and hydrophobic. So oxygen is here, and we're just going to look at water for right now. Oxygen is very electronegative. It pulls in electrons. Okay, it doesn't totally steal the electron from hydrogen, but it kind of shifts the orbit of the electrons toward itself. So it makes itself more electronegative. Whereas hydrogen, being a proton, becomes more electropositive. And when you put these water molecules together, they attract each other, and that attraction is pretty strong. It's called a hydrogen bond. If you lower the temperature of water, that bond becomes very hard and water becomes ice. So each water molecule attracts other water molecules, always from the negative area to the positive area, not a negative to a negative. When you look at fresh car wax and when water beads on top of it, it forms a nice curvature because it's actually pulling in toward itself. And if you put two of these drops of water together, they become one really quick because they are attracted to each other. And so water has a unique property in that it attracts in every direction, but if it has a surface, it can't attract up this way. So it kind of turns itself and makes this surface area. And in that surface area, you can even take a metal paper clip and float it on water. This bug actually weighs more than water, but that surface area is pushing up on that bug. That's important because there's areas of collagen that are hydrophobic, and it's going to push it away. And there's areas of the 4-PBA that are hydrophobic, and it pushes it away. So there's areas that are hydrophilic, that's attracted to, and areas that are hydrophobic that it pushes away. So carbons are not very electronegative. They don't pull electrons very hard. So the carbon chain and the phenyl group are both nonpolar and hydrophobic. Whereas the carboxyl group has oxygens that pull electrons very strongly and make them polar, and that sets up a hydrophilic attraction to water. This is very important because the carboxyl group can shield various charges on amino acids, and when water pulls on those charges, it can pull the collagen molecule apart. So this is my molecule of 4-PBA. On my right, you have the phenyl group. Every other carbon has a double bond. And then you have the 4-carbon chain, 1, 2, 3, 4. And then it comes to the carboxyl group with a double bond at the oxygen. And another oxygen I put with a free electron right here. And you can see that it can either stretch out or the carbon chain can be manipulated and be very close together with the rest of the molecule. And that's important so that it can interact with various types of amino acids and shield their charges. Individual strands of collagen are similar to these two toothpicks in that water pushes them together. And they also have van der Waals forces that keep them together, which are actually weaker than water pushing them together. So the collagen triple helix is made of a GXY repeating sequence. G stands for glycine, which is a very small amino acid. It just has a hydrogen for the R group. And X and Y can be any amino acid. A lot of times it's proline and hydroxyproline, and sometimes lysine and hydroxylysine. And when you look at the structure of collagen as a triple helix, it's intertwined just like a rope. You'll have two 4A1 sequences and one 4A2 sequence. Always the glycine is in the center. And that's because it's small and it's hydrophobic. It's afraid of water. It doesn't like water. And sometimes the X and the Y are hydrophilic because they've been hydroxylated or they automatically have charges with them anyway. So if it starts to come unfolded, 4-PBA is coming to the rescue, and the phenyl group will go toward the center, and the carboxyl group will go toward the outside where water is. So this is similar to my triple helix, the yellow being a 4A1, and a black being a 4A2 chain. And it's going along, folding nice and neat. And all of a sudden it got to a mutation which changed glycine, which should be on the inside, to an arginine. And the arginine is not only big and bulky and can't get in there, but it also has a charge, and the charge is significant because water pulls on it and starts pulling this molecule apart. And it can unravel, and then it can react with or interact with other collagen molecules, making a huge aggregate in the endoplasmic reticulum. Well, along comes 4-PBA, and here's its phenyl group and its carbon chain and carboxyl group, and the carboxyl group can interact, especially with other charges. So it can interact with this, it shields the charge so it's not pulled as hard by water outward, and then the blue area, the phenyl group, can kind of act to bring it closer and kind of stick this thing together like peanut butter, and it starts help folding that together and helps it fold. So then once it makes it over that bump, it continues to fold much easier because it's trying to get to its natural lowest energy state or Gibbs free energy state, and it will keep on doing that, and with help from chaperones in the endoplasmic reticulum. And in specific, one of them holds it together called heat shock protein 47, which we'll talk about. There are many enzymes that help stabilize the collagen molecule so that it comes out into the basement membrane very solid, binding integrins that can hold it to the cell membrane, and the 7S region binds to itself, and the NC1 region can bind to 3 other collagen molecules. In this scenario here, there's no mutation. The 4A1, and 4A2 form a nice molecule. It goes in, it has cell signaling, everything works perfect. But in the next one, you see a mutation. It's toward the end of the molecule, luckily, because if this was all frayed out, more of it would be able to react with other unfolded proteins in the endoplasmic reticulum. And that would even lead to more accumulation of aggregate proteins in there. So if it does make it out into the basement membrane, we don't know for sure whether it works properly in all the cell signaling that it should have, and we don't know if it has all the binding that should take place. In this scenario here, it not only didn't make it out, but it becomes deficient in collagen. So one thing that I look at is, can we make normal collagen always come out? Can we make mutated collagen come out in a way that it's properly hydroxylated and glycosylated and folded and act as perfect collagen? And can we get rid of collagen that is not going to be perfect and proper in its interactions in the basement membrane? Can we send that off to the proteasome? And how do you do it? So in a normal cell, DNA in the nucleus codes for messenger RNA, which comes out to the endoplasmic reticulum and is translated into protein. And if everything works right, it goes to the Golgi apparatus and is finally excreted out into the extracellular matrix. However, if it's not folded properly, it aggregates in the endoplasmic reticulum, making the endoplasmic reticulum very stressed. And you can see that by the unhappy face. Or if you want to measure various enzymes that go through the roof, that's actually more scientific. So these protein aggregates can build up, and the endoplasmic reticulum in Gold syndrome patients tend to be 3 to 9.3 times normal size. And if the stress is not relieved, the cell will die. It actually gets sent into apoptosis. So there is a mechanism here that actually helps to relieve stress by cutting down on the number of proteins that are being produced and increasing the number of chaperones that help fold the proteins and also help get those proteins out over to the proteasome to be broken down, and the amino acids are then reused. Okay, this paper by Murray et al. Out of Dr. Tom van Agatmael's lab in the UK is a very cool paper. It shows you a father and a patient that have a collagen 4A2 mutation. The patient has a hemorrhagic stroke and definitely has symptoms, but the father, they say, is symptomless. And the two things we're going to look at is PDI and the collagen, the mutated collagen, to see if it gets out. So PDI is protein disulfide isomerase, and it resides in the endoplasmic reticulum where the protein is made and is folded. But it is there to make disulfide bonds in between the trimer. So it is upregulated when there's ER stress and when there's unfolded proteins. And in this, you can see that the patient has a lot of it. It's red. And the collagen 4A2 is green. The father has some increased unfolded protein stuck in the ER, but the patient has a lot of that unfolded protein stuck in the ER. And a lot can lead to apoptosis. ATF4 is upregulated in response to unfolded proteins, and it is part of the unfolded protein response mechanism. When you see a lot of it, you know that there are unfolded proteins in the endoplasmic reticulum to the point that there is some stress on that organelle. What's really interesting to me in this is that when I look at the wild type, this basement membrane is nice and thick. It's relatively even. When I look at the father, it's pretty thin in most areas. And when I look at the patient, it's comparable to the father. So the question remains, is the basement membrane being made good enough in the patient, or is it ER stress, endoplasmic reticulum stress, that is causing the patient to have more symptoms? So in my next video, we're going to look at the UPR markers for finding therapeutic doses of pharmaceutical chaperones. Then we will go through the Unfolded Protein 101 mini course, and we'll see how the UPR applies to Gould syndrome or collagen 4A1 and 4A2 mutations. We'll look at the known markers that can be found in serum from the UPR and apoptosis and other diseases. And then we're going to consider what other unknown markers could help with therapy. Thanks for watching. Give me a thumbs up, subscribe. Check out my website at the end of October of 22. I especially want to talk to researchers and clinicians about further experimental studies and clinical trials. Thank you so much.